Let me come over so that I can hear you. No. Basically, all they say is that OOT is the same as an OOS. Yeah, you still have to investigate it, just like you would an OOS.
Okay.
That's the main thing.
Okay. My second question is that you see in the OS there is two investigation, laboratory investigation as
Right.
well as process investigation. Suppose you are unable to find any any reason in the lab investigation as well as the process investigation. So what will be the fate of the batch?
Okay, if you go through, okay, you do your lab investigation and you cannot find an assignable cause.
Yes.
You do your full investigation, and again, you still can't find an assignable cause. At that point, what you need to do is you do a risk analysis on that batch. And you say, what if, okay, this is a true OOS or an OOT or whatever. What is the effect on the patient? You know, you see, this is, okay, there's a problem here. If you can argue that you can release the batch, then the question is that don't you have, aren't your specs too tight? Okay, but you know, having too tight specifications actually is not a problem. FDA likes that, except that it creates problems for you. But you have to do kind of a risk analysis on that batch to say, what are the problems here? If we release this batch, what could happen? And remember, there's a risk analysis to the company here, which is you release this batch in the face of a known OOS, and you're saying when you release that batch that it still should not be dangerous to a patient. The problem is that you release this batch and some patient out there has an adverse event. And then their lawyer comes back, does an investigation and says, you know, you guys released this against a known OOS. It's negligence by the company. At which time you are going to have to go to court and defend the decision to release that batch. So that's the only thing.
My second question, in the blend informative, right? Suppose you get one result out of 10 result is okay, one result is out of spec.
Yeah.
So, and then you investigated that there is no laboratory error. Okay.
Right.
In the process, also you investigated there is no reason to find one result is out of spec.
Right.
So can we go ahead for the compression? And if the compression found okay, all the tablets,
yeah.
so can we release the batch? Though,
yeah.
though there is a failure of one
if,
blend informative.
If you think that there is no reason to believe that there's a blend problem, and you go and you compress it, and then you show that your content uniformity is good. And what I would do here is I would increase the number of tests. But then if you say that the content uniformity is okay afterwards,
yes.
It's acceptable.
Yeah. Sir, regarding stability, suppose stability samples you are getting the OOT result, stability. Right. So definitely you are going to investigate as per the lab investigation. So is it required to go back to process also? That is called part B investigation in the case of stability.
Yeah, stability?
Yes. You will do part A investigation.
Yeah, well, here's the thing. A stability failure automatically creates an OOS.
No, okay. not failure, it is OOT. It is
okay, OOT, yeah. Well, any of those failures
yes.
must be investigated. Now, you have to assume that it was a process problem that led to that. Okay, so you do need to go back and look at the process and find out what could have caused the OOT.
Means you have to go back to process also.
Right. Now, you see, the problem here is this, on stability sampling. You may have a product where you know the thing is degrading. And in fact, that might be your standard trend. That you say, well, I have this thing, and over a period of 3 years, I know I get this slow degradation. But this whole thing is within the acceptable range for my product. And then the thing goes OOT. It suddenly drops fast or something like that. You have a problem here because according to the behavior of that trend, you had a normal product, right? Up until the time that the OOT took place. And I will tell you, most of the time, you have either a sampling error or a lab error. Because the product was acting normally until that OOT occurred. So, at this point, really take a good look at the possibility of laboratory error.
Yeah, sir, one more question. In the case of the OS, during laboratory investigation, you found that there is a assignable cause in the laboratory, and then you are going to rectify that cause.
Right.
Right. After rectification, so the analysis will be done again. So is it one analysis required or 5 analyses required? Because there is confusion.
No. If you find an assignable lab error,
Yes.
that invalidates the test.
Right.
So when you do the retest, you don't have a previous test. You start it all over fresh.
I know, sir.
Yeah.
After rectification, you have to analyze one assay. Suppose it fails in assay, you rectify the problem.
Right.
So again, when you are doing the retesting,
When you retest, no.
Same sample.
You see, it's not a retest.
Same sample analysis.
Yeah.
Same sample.
Same sample, same everything.
So is it one sample preparation
Yeah.
or 3 or 5?
Just one. Whatever your normal SOP was. Okay. Think of it this way. When you find that the original test had a problem due to an assignable cause, you've just invalidated that test.
Right.
So when you do the retest, you're actually doing the first test. You're not doing a retest. That first test does not exist. So just follow your normal SOP.
And this OS is also applicable for packaging material?
Yes?
Okay. And last question now, you see, like you're doing the FMEA. So this FMEA is for process or for particular product? Like
Okay, you can do it both. Well, in fact, you should do it both ways. Yeah, but it is for a particular process.
Okay.
And then if you're using that same process, say, to make another product, okay, if you can assume that that product behaves the same way as the other product, then the FMEA is still good. But if the mechanical properties of the new product are somewhat different from the other product, then you're going to need to redo an FMEA.
Okay, thank you.
Okay.
Thank you, sir. Thank you.